All right, so I'm really excited to be here. We already heard two amazing talks. It's really hard to follow those two, but I try my best. And I'm really happy that I came after Yushan and Eva because they both covered parts of the things I want to talk about, so it makes my life much easier. Today I want to talk about computational approaches to design peptide binders, and mostly what you hear from me are the challenges that we face and the problems that are on the way of approaching this. So let's get started with a motivation slide, which I think is totally unnecessary for this audience over here. But we all know that a huge space of the disease targets is undruggable. When you look at these undruggable targets, many of them are inside our cells and have flat surfaces. Just to show you what I mean by flat surfaces, I'm comparing an enzyme beta-lactamase with protein that binds to the receptor PD1. Here in pink, you can see the substrate binding, footprints of beta-lactamase substrate. So you can see it's like a nice small pocket. Whereas when you look at how PD1 is interacting with this receptor, it's a really, really large interface. And these two are drawn in size. So like you can actually compare the scale. So it's really huge, it's really big. And they're both inside the cell. So why does it matter? So if we look at the two common modalities that people use for therapeutics, none of them can access these flat surfaces that are inside the cell. Antibodies are amazing at binding to things, but they're too big to get into the cell. And then on the other extreme are traditional small molecules. And I use the term traditional small molecules for those that follow the rules of 5, because more and more we're seeing small molecules that are being discovered that are very big and are not obeying any of those and are working great. But the traditional small molecules haven't been amazing at binding to those flat surfaces with the selectivities we want. So somewhere in between are peptides, what we all like. They are able to enter the cell. At least there are peptides that we know can go inside the cell. They have the ability to bind to flat surfaces, much in the same way that antibodies or other proteins are binding. And in fact, many protein-protein interactions are actually directed by these peptides or peptidic fragments. And there are natural precedents of peptides that are used as therapeutics or are being used in our bodies for defending our body or as hormones for signaling and things like that. So they're really cool. They hold this promise of being this complementary therapeutic modality to target these flat surfaces inside the cell. And people are working a lot on obtaining peptides. And library-based screening methods are the ones that have been used with huge success in getting binders. I'm just showing you one example of a library-based screening method going from a DNA to RNA. You have a promycin moiety here. So when you start translating your mRNA, your amino acids get attached to the mRNA. You do some sort of binding screening over here, you get your binder, and it's really, really powerful. But like any experimental technique, it has its own limitations and challenges, right? One limitation is that most of the time you have no control over where this binding happens. You can only say that things are binding. And then you have to do some downstream characterization to figure out if it's binding in the right spot for the right reasons or not. I think the bigger challenge is actually, despite the fact that some of these library-based methods can screen many, many peptides, still the potential space of peptide sequences is just too vast to be sampled with any experimental techniques, right? I'm showing you a potential space of an 8-mer that's built out of L and D amino acids. And you can see it's very quickly hitting 10 to the 12 possibilities. And if you think about the mRNA libraries, they're usually 12 mers, so much, much bigger. And this space can rapidly grow, right? You can use unnatural amino acids or non-canonical amino acids. And by just adding 10 unnatural amino acids, this space gets to 10 to 14. If you add N-methylations also, you have 10 to the 15 possibilities. So like space is huge. The largest libraries we can build is 10 to the 14, assuming everything goes well in your library production, right? So why is this bad? Let's say you have this entire space, your library-based screening methods only sample this much, and these orange and yellow dots are things that bind and are potentially good hits. Many of them need to go through downstream optimization to get to the pharmacokinetic properties that you want. And these downstream optimizations can fail. A lot of the times they do fail. So you're missing a lot of these other potential good binding partners that you just don't have in your library. So the whole idea behind computationally guiding this design is that can we, instead of just focusing on a small part, guide these libraries to say, hey, sample this part, and this part, and enrich the libraries with the good binders so that you don't miss out so much. And then hopefully narrow down the time that you require for downstream optimization. So that's a project I started from my postdoc. And the first step in making this goal possible was to show that we can control the shape of the peptides or the structure of the peptides, because the structure directly correlates to the binding and the function. I'm not going to discuss that, but because it's published a while ago, but we showed that you can actually design peptide structures with high accuracy and with full control over its shape. So there are ways to create peptides that are fully structured. And when I'm talking about peptide truly stacked, I mean cyclic peptides that are constrained in two ends, right? So just for a brief overview, I'm showing you NMR ensemble over design, and you can see that they work really nicely, and the RMST is really low. Only thing I want to mention from this paper is very quickly talking about the method, because it becomes relevant later on when I talk. So the method we use for this is called generalized kinematic loop closure, and I'm going to refer to it as KIC for simplicity. It's inspired by robotics. So let's say this is a robot arm in 2D, and you want to close the two ends. What you do is that you randomly move it around these degrees of freedom, right? And around where the elbow should be. And you can bring the two ends together. You can think about an amino acid side chain much of the same way, where each amino acid is one arm of this robot arm. And then instead of moving things in 2D, it basically moves in 3D by changing the phi and psi in the torsional space of the peptide. And then you can, with this method, you can rapidly sample tens and tens of thousands of conformations. Some of them will not work, but many of them work. So this is a stochastic random process that can sample lots of conformation for you. And that's all that you're going to hear about structure design. The next step is to actually generate a peptide binder. One of the big questions that we try to answer is that where do we start? How do we make sure that our peptide that we design bind to the surface that we're interested in? Because that's one of the problems you see in the library-based screening methods. After checking multiple different approaches, what we came up with is this anchor-based approach. It's really inspired by what people before us have used to steal like a motif or a loop that you see in nature and close it or staple it. Here, instead, we're using just the one or two functional groups, and that's it, not anymore. And then create the peptide around it. Here, I'm showing you an example of one of my recent works on designing peptides that can bind to and inhibit histone deacetylases. So the main anchor here is this long unnatural amino acid, which is basically a super long cysteine that coordinates to a zinc in the active site. After you find your anchor, either from a crystal structure or your amazing chemical intuition, then you extend it, add more amino acids around it, computationally, of course. And then you use the KIC method that I explained to sample all of these closed conformations in the context of the protein. And it's really important that the protein context is around, because you want to kind of bias your sampling so that you make more interactions with the protein surface. Right now, there's no amino acid side chains assigned to it. So the next step is to go through sampling all the possible amino acid side chains, assign some, and then generate peptides. And this process is very fast. So you can generate tens of thousands of peptides in silico. And the problem really becomes which one to choose, right? So we have a bunch of computational interface metrics that we use. You don't need to really understand this slide. This is just like showing correlations between a bunch of these different metrics. And I just wanted to show you that you have to consider many, many different things, because from what our experience shows us, none of these correlate with binding exactly. But all of these throw out the bad peptides. So you have to check all of them and put thresholds for them and throw out the bad ones. And you get a subset of good ones that bind relatively well. So how did we do with the histone deacetylase that we were working on? We chose 50 peptides based on the thresholds that we picked. And we managed to get peptides with different binding affinities. So here in pink, I'm showing you the original anchor. It binds with 5 macromolar affinity itself. Most of our peptides have IC50s that are lower than this. So I call it a success. And note that we only test 50 instead of thousands of peptides in a library. And the best binder binds in with 9 nanomolar affinity, which is, I would say, pretty good for histone deacetylase 2. And we have one that binds with 4 nanomolar affinity with 2 histone deacetylase 6. So that's awesome. The method works. We now have a pipeline that you can walk through, going from a structure to a binding surface, to an anchor, and then to a peptide design pipeline. Things are not always this easy, because what our pipeline also showed us is that there are a lot of challenges that we have to face. And I'm going to walk through some of them. The first one is that you need to have an anchor. So you can't just start with a surface and put your peptide in. So this is one of the projects that my lab has started to think about how to solve. We don't have any awesome smart solutions to it. We know that we can use the information we have from PDB, from proteins that are interacting with each other, and how protein folds to actually correlate surfaces with functional groups. But this project is really in its infancy. So I'm just going to leave it up to here and put it as a question to the audiences. How can I start with the surface and know what functional groups work in there? The other big problem that we noticed was that when we design our binder, we get a binding orientation and we calculate all our computational metrics in that binding orientation. When we got crystal structures of our complex, we got very surprised because we noticed that our peptide actually did rotate in the binding pocket and made completely different interactions from what we designed for. So this is a big problem because we can't really use our computational metrics to correlate them to binding anymore, because they are on 2 very different structures and binding orientations. So why is this happening? There's probably a lot of reasons in here, but the main reason from what we've seen is an undersampling of conformations. Our design focuses only on 1 conformation in solution and 1 bond conformation. And our idea is that if we optimize the energy of these 2 enough, we don't need to worry about anything else. But that's really hard to do for a peptide. Peptides are so tiny that conformational changes in solution doesn't really have a high energy barrier for most of them. And also, like, the changes that they have in the bond conformation can have really huge differences in binding affinity. So really the question that my lab's trying to address is how can we get a better understanding of the conformational ensembles in the unbound form and also in the bound form? And can we use this information to finally get to a computational metrics that generates binders, that create binders in the exact right orientation that you see in the experiment? Well, good thing is the method we used for designing peptide actually at its core is a fast conformational sampling approach. It doesn't do design, it basically samples all the conformations for you. So you can run this method, you can get a bunch of conformations, you can find which one has the best score, and we use Rosetta score because that's the software we use. And then we usually use plots like this. And the y-axis, we have the energy. On the x-axis, we have RMSE to best design. And then we generate tens of thousands of conformation within 10 to 12 hours. And the cool thing about this plot is that it's actually a compressed version of the peptide energy landscape. So like if you think about the 3D energy landscape and you compress it such that things are just RMSE and energy, this is what you get. So really the project that we're working on right now, and it's inspired actually by Yushan's work, is that can we combine this sampling of conformations with molecular dynamic simulation to get a full picture of this energy landscape and recapitulate that's this 3D funnel shape. And that can help us know what are the conformational ensembles in solution. So pipeline is very simple. You get from a sequence, you do tons of sampling, you do clustering because these are stochastic and redundant. And as Yushan said, many of them have very similar shapes, you know? So we do our own clustering method. And then from here, we run molecular dynamics simulation to find transitions between them and like try to sample more of the space. Does it work? It seems to be working fine. And it seems to be actually helping with conformational sampling of the space compared to normal MD. My white box moved up. So this is the REMD, replica exchange MD simulation, starting from the native conformation. So this is the native conformation, and you can see that there are some other conformations that are popping up and we're sampling them. What happens if we start from the conformations that we created using Rosetta-based sampling instead of just the native conformation? On the same plot, you can see that starting from different conformations actually help you sample different parts of this conformational space, and you can see, I guess, more of this energy funnel that's kind of missing, right? So this is here, so we saw that before. This is basically here, but this is new. You don't see that here. So this is like a trapped conformation that REMD is missing, but because we sampled that conformational space, you get to see that over there. So we're really excited about this, and we're hoping to use this to gain a better understanding of peptide conformational ensemble in solution. And we're also asking the question of, with these conformations that we get with Rosetta, can we run or replicate similarly to what Yushan did with MD conformations to be able to generate a predictor? And since this is really fast, we can generate conformational ensembles for tens of thousands of peptides at once. So we don't need to, our data set can be huge. So can that help with getting a better understanding of this? So this is one of the main areas of focus in our lab, and we're quite excited about how it's going. Another challenge that peptide therapeutics usually almost always face is the permeability problem. You get a peptide, you're excited, it binds really well, either from your library screening or your design, and then you end up being outside the cell. And you're like, okay, what can I do? Because my target is inside the cell. And there's a bunch of downstream optimizations you have to go through to hopefully get one of your peptides crossed. A lot of the times it fails. A lot of the time it interferes with your peptide ability to bind. So if only we could, as a community, come up with a better predictor for peptide permeability or ways to know which peptides can cross, that would be super helpful. But it's really hard because, first of all, there's so many peptides, and you can't test all of them for permeability. Most of the assays don't have that high throughput to be able to test everything on. So for example, library of hexameric cyclic peptides, just from the sequence, have 10 to the 25 potential sequences that needs to be tested. And the number of data that's available for machine learning is not a lot. And that's like the region that Eva was talking about. So this is not really a deep learning region that you can use, maybe a machine learning region. One of the challenges with it, there's been tons of studies on permeability, especially passive permeability. The main problem is that they all focus on small subsets of this space that are very similar to each other. So like maybe mutated variants of 6 mers or similar variants of 7 mers with different patterns, you know? And the diversity really matters because what you learn in here really can't be generalized outside of here. So really, the pipeline that we think could help is to find a representative subset. It doesn't have to be huge. I guess 6 can be enough sometimes. I couldn't expect that, but that's amazing. It doesn't have to be huge, but it needs to be representative. It needs to show what the entire space has so that you can learn the underlying patterns. So the pipeline that we came with is to find a way to generate this diverse subset and then test them and build a machine learning. It's not going to be deep learning because we can't test so many, but build a machine learning tool that can then learn permeability on the bigger space and repeat doing that and hopefully get more and more of better permeable peptides. In order to create diversity, we need to know the distance between sequences, and that surprisingly has been very difficult. So when I show this to you, these are one-letter codes of amino acids. As chemists and biochemists, you have different reactions to these mutations, right? Alanine to serine, yeah, okay. Alanine to tryptophan, not sure. Tryptophan's too big, you know. Proline to anything, oh, let me think about it, you know. So you have a sense of what is happening and what shouldn't be happening. We were looking for a distance matrix that captures our biochemical intuition of these mutations. As has been difficult, we tested many different matrices, distance matrices, and I would say fingerprints of amino acids that are out there. And the one that worked really well for us was VHSE8. So it's the principal component, the score vectors of a bunch of biochemical properties and biophysical properties of amino acids. So the way we define the distance is very simple Euclidean distance metrics given this VHSE8 on the 8-dimensional space. And then we come up with a threshold for accepting mutations, and it works really well. So isoleucine is most likely going to be isoleucine. If not, leucine or valine are the next choices. So it intuitively makes a lot of sense. And if you look at all the amino acids across the board, the potential mutations that it's suggesting is meaningful. So using this, we managed to kind of cluster our space and find different distances. Clustering is not really the right word. So the space, as I mentioned, is like 10 to the 25 sequences. So many of the methods we thought we can be using on it just didn't work on a space this huge. So what we ended up doing was to start with a sequence, random sequence, and then randomly sample other sequences and just make sure that the samples that we get passes a distance threshold, so it's diverse enough. And it converges after like 250 steps. So surprisingly, there's not a lot of patterns in the space. So my student, Noura, what she did was that she tried to kind of come up with a code that combines the features of the space together. So how many polar residues are in the sequence, how many charge residues are in the sequence. It's just like a very simple feature code so that we could group these sequences. And then she started from many different sequences, combined and collected all of these and looked at these features in them and saw how many she was collecting. So all these blue dots, blue lines over here are starting from a different starting sequence. And she's just stacking them up so that you can see them. All of them actually start from 0. So all of them follow this red pattern. And the cool thing is that the red one is actually what you get if you calculate the probabilities for each of these groups. So a random sampling is, in fact, truly representing the groups that you see in the sequences. So we picked 96 peptides from these groups that we get. They're all synthesized, and we'll be testing them for passive permeability with PAMPA and also KCO2 permeability. And we hope that we get really cool results. They're going to come in 2 weeks, so we'll see. And yeah, that's it. Hopefully I'm in time. I usually talk really fast, so I think I'm in time. And with that, I want to thank my group, especially Noura over here, who's done all the permeability work, and Sushtana, whose picture I showed, she's not here, who's doing the molecular dynamics simulation combined with Rosetta. I would like to thank the funding, University of Oregon Knight Campus. Thanks to the organizer, and I'm happy to answer any questions. Thank you.